Okay, hi everyone. Let's today take a closer look at B cell function, how B cells properly are able to see the antigens that their immunoglobulins are designed to recognize, how they are encouraged to interact with T cells and other immune cells in order to properly activate and carry out their functions. Um, and we will examine the role of certain immune cells that are in the lymph node, whose job it is to coordinate the activation of B cells and T cells and get them working together. As a quick reminder, before we start the lecture, you do have a quiz due this week. The quiz is on the T cell material from chapter 8 that we wrapped up on Monday. And so as usual, your quiz is due Thursday night, tomorrow night by 11:55. All right. So this class as a whole has done a very good job of keeping up with the quizzes. So I hope you guys, keep that up. All right, that's good. So don't forget to take your quiz. All right, let's begin by just kind of thinking about comparing, natural killer cells with B cells and T cells. We've done this kind of on and off as we've learned about these closely related lymphocytes. But I think it's really important that you have it clear in your mind why it is that we have these three closely related types of lymphocyte cells that are, obviously part of our adaptive immune system, to keep them sort of separate in your mind about what makes them similar, but what makes them different, right? Because it's really important that you can be comfortable understanding how these different cells work together. So let's start off with our natural killer cells. So the natural killer cells, right? Oftentimes I've said that even though they sort of represented evolutionarily a separation from our innate immune effector cells, remember, for eons and eons, multicellular organisms have had a innate immune system, because you have to have at least an innate built-in ready-to-go immune system to be able to deal with sort of common everyday pathogens, right? But those innate effector cells, things like the macrophages, the neutrophils, even in the granulocytes, like eosinophils, basophils, which we will talk about more later on, those are always concerned with just kind of targeting the pathogen directly, whether it's through phagocytosis, like eating up bacteria or viruses the way that macrophages and neutrophils do, or things like releasing certain types of noxious or nasty type of chemicals, compounds from vesicles, the granules that come from inside of things like eosinophils, basophils, and mast cells. They're done mostly in a very non-targeted kind of way, right? These cells activate and they just release what they have inside of them. So we said that the natural killer cells, evolutionarily, were kind of like a departure from that, right? Because natural killer cells, remember, they're not concerned with, finding a bacteria and somehow eating a bacteria or virus or whatever, right? Natural killer cells are more concerned with going around in our body and checking out the cells of our body that it bumps into and seeing if there's anything wrong with that cell that might indicate that the cell is diseased or old and needs to be recycled, or possibly contain evidence of infection. And then the natural killer cell will kill that target cell through releasing cytotoxic types of signals, small little cytokines that we think of as cytotoxins, because the result of those cytokines being received by a target cell is that the target cell undergoes apoptosis and self-destructs. Right? So we said that even though the natural killer cells are different than things like macrophages and neutrophils, because the natural killer cell relies upon talking to or contacting other body cells and evaluating the status of the other body cells, it still functions in a way like an innate effector. Why? Because natural killer cells just come preset with a selection of different types of protein receptors on its surface that are coded for by your genes, right? And these are the certain types of proteins and genes that over many, many millennia of evolution turned out to be very useful, right? So again, that's why we consider the natural killer cell still functionally part of our innate immunity, right? It cannot adapt. It cannot learn anything new. It just does what it does. So that's why the next step then from the natural killer cells evolved the T cells. So the T cells, right, not only have an assortment of different receptor proteins on its surface, just like the natural killer cells. Remember, the T cells and the natural killer cells are from the same sort of precursor cell in your bone marrow that then gets released. And some of them will become natural killer cells. Some of them will make their way to the thymus where they become T cells. So really, a T cell is like an upgraded natural killer cell, right? Because once in the thymus, the T cell or the thymocyte, right, learns how to make a T cell receptor in addition to the other receptors that it has on its surface. And as we've learned in this class, the process of making T cell receptor using VDJ recombination is to create a multitude, right, millions upon millions of different shaped T cell receptors, some of which you hope will be the right shape to actually be useful in detecting a novel pathogen, right? So this is the difference now. The T cells still go out and associate or synapse with your body's cells, right? Just like natural killer cells would do. But whereas maybe a natural killer cell would fail to detect something on the surface of this body cell because the preset receptors on the natural killer cell, did not come pre-made to recognize a brand new type of peptide signal from a brand new virus, right, that our species has never seen before. Hopefully you'll have natural killer cells that, because they have unique T cell receptors, right, it's the T cell receptor that might recognize something new. And therefore you select that T cell and you want to make lots of that particular T cell. And we said that you have two types, the helper T cells and the killer T cells, right? But still, right, they work by contacting a target cell and, depending upon what they see, send cytokine messages, either cytotoxic messages to kill that cell or helpful cytokine messages, like a CD4 cell that helps some other cell like a B cell. So let's talk about B cells then. Right? So if we went from the evolution of natural killer cells to sort of an improved version of a natural killer cell that now has this T cell receptor on it, so those are T cells, how does the B cell kind of fit in? The B cell is also, in a way, like an improvement on the T cell. Because the T cells, you say, great, I have these novel T cell receptors on the surface. That allows me adaptive immunity, meaning I can select for T cells that happen to have the right shaped T cell receptor that will allow me to be able to detect and fight against a novel pathogen. But remember, the limitation is what? T cells can only detect small little peptides with their T cell receptors. And the other kicker is those small little peptides are not peptides that are just floating by, right? Those peptides have to be presented by the other cell that the T cell is synapsing with, presented on MHC platforms, so that a CD4 cell can properly recognize MHC2 presented peptide, and a CD8 cytotoxic T cell can properly recognize MHC1 presented peptide. And that way you control what happens next. But, you're still limited by the fact that the T cell receptor is permanently stuck to the surface of the T cell, right? And it's stuck only being able to evaluate small peptides on MHC. So the next evolutionary step you can think about is, hey, wouldn't it be great if we could make a similar type of antigen-specific molecule that isn't stuck to the surface of the cell and isn't limited to only receiving peptide? Hey, that's what an immunoglobulin is, right? So B cells are like the latest invention of our immune system. I use that term kind of funny. You're like, latest invention, whatever. How long have we had that? It's not like we came up with this last week, right? But in evolutionary time, B cells have only been part of complex animals, vertebrates, for something like, I don't know, like 200 million years. All right. And you're like, that sounds like a long time. Well, but when you consider something like innate immunity has been a part of living things for billions of years, right? 200 million is pretty recent. Right? It was really just part of the vertebrate evolution. So in B cells, you can think about it this way, that T cell receptors evolved into immunoglobulins that allow non-peptide specificity, right? An immunoglobulin can bind anything. Yeah, it can bind a small peptide, but it can bind carbohydrate, it can bind fats, right? Other things, or bigger protein, anything that the little immunoglobulins antigen binding arms are the right shape to detect, right? And remember, the whole reason why immunoglobulins, have the light chain, heavy chain, and if you look at a T cell, the T cell just looks like what? Two chains together, like one arm of the immunoglobulin. That's not a coincidence, right? Because the immunoglobulins sort of evolved from the original T cell structure and improved upon it. Now you have like two of them, and you have them on a stem, or you have a Y-shaped molecule that can then be released by the cell. And that's a big deal, right? Because once you talk about B cells can be chosen because their immunoglobulins right shape and encouraged to become plasma cells to release tons and tons and tons of free-floating antibody version of their immunoglobulins. Now you've really ramped up what your immune system can do, right? Because now the cell that makes this immunodetecting molecule doesn't have to be near the actual target, whether that target's a bacteria or an infected cell or whatever, because the immunoglobulins, the antibodies, can just flow through your body and do their job, right? Independent of being attached to a cell. All right. And you sort of go back now to being able to target the pathogen itself, right? So if it's a little bacteria or a little virus, antibodies can now on their own target that pathogen directly. So it's kind of like what comes around goes around, right? Our innate system evolved, billions of years ago to target things like virus and pathogen directly. Then the adaptive system came along to sort of say like, well, let's actually also pay attention to any cells in our body that are already infected by this, right? And then you have the B cells, which take that really to its sort of logical conclusion, which is, yeah, now that we know how to make these specific antigen binding molecules, right, the fact that we can release them now enables them once again to directly target pathogen like bacteria and viruses. So that, I want you to think about that, right? Because I think it's wonderful to be able to kind of put that all in perspective. And if you sort of understand that, then you understand the relative contributions of these different types of cells and how they're similar and how they're different. All right. Let us revisit the anatomy of a lymph node. We've seen multiple cartoons, simplified cartoons from the book of a lymph node, but this is a nice one that's a little bit more anatomically correct. Right, so remember, lymph nodes are sort of a classic example of secondary lymphoid tissue. This is where lymphocytes go to actually do their job, right, where they go to activate. And remember, the reason they work so well is because they represent places where immune cells and antigen can be brought together in higher concentrations to encourage things like lymphocytes to be able to encounter the antigens that their antigen-specific molecules like T cell receptor or immunoglobulins actually recognize, right? That was the example of that's why when you want to go buy an iPhone, right, you don't go wandering through a flea market hoping to find somebody to sell you an iPhone. You just go ahead and go to Ala Moana and head right to the Apple store, right? Well, actually, maybe you don't, because I think that's like the worst way to buy an iPhone, because then you pay like $1,000 for it or something, but whatever. But the point is what? By having it localized somewhere, right, you know to go there to be able to find what you're interested in. And right, the salespeople in the Apple store, you know that they are the actual real salespeople. So any type of conversation we have with them, you feel like is more reliable or trustworthy than just some random person who has a table at the Aloha Stadium flea market, right? The reason I wanted to show you this again is because remember, I'm like hung up on this idea that students get confused about this high endothelial venule, right? So this is how it's drawn in some of the book's cartoons. And remember, this is the place where lymphocytes that have been going through the blood end up exiting out of this into the lymph node. so that they have a chance to check out what's in the lymph node. If they don't like anything that they see there, they're now part of the lymph system. So they would then drain out of the lymph node and be part of the lymph system until they eventually get reintroduced into the blood again up at the level of your shoulders, right? But remember, I told you that the way the cartoon is drawn, it's kind of misleading, right? Because it looks like this magic, like floating donut, right? This purple donut. I don't call it the magic purple floating donut because I've made that up, right? I mean, I actually had students in previous semesters say, you know, oh, the high endothelial venule, that's that magic purple floating donut, right? That goes bouncing around inside of a lymph node, spewing out lymphocytes, right? And I think the first time a student told me that, I just kind of like stared at them. Yeah, but anyhow, so I wanted to emphasize, right? High endothelial venule is a specialized region of our capillary bed. So in this illustration here, so this is why this picture is nicer than the ones in the book, right? This illustrates that you have a blood supply flowing into the tissues of the lymph node. And that blood supply, a big reason you have that is to remember, the lymph node is living tissue, right? So that means the cells in here need nutrients, oxygen, they need to get rid of CO2, waste products, etc. Right? So you have blood vessels that will enter the lymph node, and in here, so this arteriole that comes through, right, it branches off into little capillaries, and so this whole inside is filled with this capillary network, and each one of these capillary beds is composed of teeny tiny capillaries bringing oxygenated blood in, and then they kind of turn into teeny tiny veins to bring waste products, carbon dioxide out. Right? And so throughout the whole lymph node, you would have these capillary beds. You know, the illustration only bothers showing this little region down here, but that's what's going on up here. This would continue on, right? And in each of the spaces up here, you'd have this. This is nice because it shows you what? That the blood flow is continuous, right? The blood that comes in eventually goes through capillaries and then it eventually flows out. So in that blood are things like lymphocytes, B cells, T cells that have been going between blood and lymph. Here they're coming in through the blood into this particular lymph node. Right? They go through the capillary bed. And at the end of the capillary bed, as they're sort of becoming the venules, or the capillaries are becoming the venules, you have this region of sort of a thicker sort of spot. This is the high endothelial venule, right? So if you were to imagine this drawn in the cartoon as a cross section, that is why it's drawn as the magic floating purple donut, right? So in this region, the endothelial cells, the cells that make up the wall of the HEV, have those special proteins on them that act to sort of attract and stop lymphocytes that are flowing through, encourage those lymphocytes to stick to the walls and then squeeze their way out into the lymph node, right? So in that way, they've gone from the blood into the lymph system. And then from there, they cannot squeeze their way back in. That's why they are now part of the lymph system. Their only way to go and move on is to drain out of the main efferent lymph vessel, you know, onto another lymph node and maybe another lymph node, eventually being reintroduced into the blood up at our thoracic valve up at our shoulders. All right, so let's remember that B cells, they mature in the lymph nodes, right? Because the B cell that has been released by the bone marrow needs to undergo tolerance training in the blood to make sure its IgM doesn't cause any trouble by binding to things that would be, you know, kind of conservatively assumed to be normal human body signals, right? Antigens that really are self-antigens, and that these T cells should not be allowed to go to work if they actually bind to self-antigens, right? When they get into the lymph node, remember, they are rewarded for their good work, right? So the naive B cell that's still immature enters through the blood. So the blood vessel will come up behind here in this cartoon. Then here you have the high endothelial venule. That B cell makes it into the lymph node where it is rewarded, right? Rewarded by big follicular cells in the lymph node. These are our follicular dendritic cells that then help the B cell mature. So now it's IgMs on its surface if they go on to bind something now the B cell will be stimulated. Right? And then I showed you this slide before, but I always think this is funny, this last cartoon here, right? I always joke this super anatomically correct illustration, but it's useful because it reminds you that now this mature B cell goes around and around between the blood and our lymphatic system. Right? So if a B cell enters a lymph node where, this B cell is mature now, right? It got rewarded in some lymph node somewhere, and now it's been going to work between the blood and the lymph, blood and lymph, for maybe many days or a few weeks. It's just been going back and forth, back and forth. And then one day, right, it has the opportunity in a lymph node to actually encounter something like a pathogen. How does the B cell begin to kind of realize that if it's binding to something that its immunoglobulins recognize, that it should be something that should start its process of activating? Because again, remember, right, just like any other immune cell that's floating through your blood, the vast majority of other things it bumps into in your blood are totally normal signals from your own body, right? Whether you're talking about small little fragments of molecules like carbohydrates, lipids, or small little peptides that are just scraps from other proteins that are floating around, right? Small little peptides sometimes actually serve as signaling molecules through your blood, like hormones, for example, right? So when a B cell does bind to something, how does it kind of know that this thing that it's bound to is not a self cell, right? That might be something like a pathogen, like a bacteria. Right? So the Ig itself is not a very strong way of binding something and holding on to something, right? Especially if you consider that the IgMs that a B cell first has, even when it's mature, right, probably is a pretty lousy fit. Close enough fit, sure, but close enough is not very strong, right? So there must be other ways that B cells can kind of really hang on and contact something so that its Igs really have a chance to detect what this thing is. So that involves other sort of generic receptors on its surface, like complement receptor 2. So complement receptor 2, as the name implies, is a receptor on the surface of a B cell that can help detect complement proteins on the surface of some other cell. This CR2, which is represented in this blue chain here, is bundled up with a couple of other proteins, CD81 and CD19, and together they make a functional B cell co-receptor. Just in the same way that we talked about T cells having a T cell receptor complex, right, as well as a co-receptor like a CD4 or a CD8, and they work together, right? Now what I'm telling you is that B cells do a similar type of thing, right? Not their Ig alone binding to something, but it's going to involve some other help from some other co-receptor. And this is how a B cell can tell the difference, that it is pressed up against a normal body cell compared to something like a pathogen, right? So let's say, for example, right, that it's pressed up against something, and that something might be just a totally normal body cell. That's you, right? Or it could be that it's actually pressed up against bacteria, right? Like bad clown, this pathogen here. Chances are bad clown is already undergoing the process of being opsonized, right? Covered in complement proteins. Right? Bad clown, though, he's so badass, though, he doesn't care, right? He's like smoking the C3b, right? Well, anyhow, so because the pathogen is covered with C3b, you have complement receptors on the B cell that can pick up on that. So the B cell has CR1, just like macrophages do, to be able to bind to the pathogen, sort of like this whole Velcro thing that we were talking about with the beach ball and the ping pong balls at the beginning of the semester. But it also has this CR2 B cell co-receptor complex. So when the complement receptor 1 binds to C3b, it encourages this other factor, factor I, which we learned about at the beginning of the semester as being sort of an inhibitory protein that actually cuts up the C3b, right? And so normally in our body's cells, we would have factor I preventing our normal healthy body cells from being suddenly opsonized. Right? When the B cell binds to C3b with its CR1, that actually encourages factor I to come and cut the C3b, right? You end up with this little leftover scrap of the C3b, which is now called C3d. Right? That is what the B cell co-receptor complex picks up on. So, in other words, if this B cell presses up against some other cell, and that cell is covered with these little weird runty bits of C3d, right? That's a very good sign that this other cell is actually a pathogen. Right? So here you see the CR2 binding to the C3d. Right? And that's how the B cell can kind of distinguish that what it's pressed up against is not self, right? That it's actually some sort of pathogen. So here you can see. So in the same way we talked about T cell receptor on T cells working with CD4 or CD8, here we have the immunoglobulin, IgM, on the surface of a B cell, right? And it detects antigen on the surface of, let's say, bad clown up here, right? Bacterial cell. But that association alone is pretty wimpy, because chances are, if this is the first time that this B cell has ever bound anything, it's just going to be like the two random puzzle pieces in the jigsaw puzzle, right? Yeah, it was close enough fit that I could pick up both by just holding on to one, but if I just shook it a little bit, the other piece falls off, right? So that's not very strong, right? So the CR2 co-receptor makes it a lot stronger, because chances are bad clown is in the process of being opsonized by complement proteins, so it'd be all kinds of little scrappy C3d parts on there, right? And that's what the CR2 picks up on. So that combined signal of immunoglobulin signaling through the other associated proteins that sort of provide a little bit more cytoplasmic domain to the IgM, right? And the signaling of this co-receptor. It happens through that LCK protein that we learned about last time that ends up being recruited here and starts phosphorylating, right? Different types of conserved peptide sequences, right? That double signal is what then sort of prepares the B cell, right? You know, they use the term activate here. I don't like the fact that they use the word activate, because activate to me means, okay, this B cell is activated now, and now something dramatic is going to happen, right? I think it's preemptive. It's like kind of jumping the gun to really use the word activate, because at this point, this B cell does not really activate. Remember, activation of a B cell requires what? Requires partnering up with a helper T cell that will then tell the B cell to really activate, and importantly, tell the B cell what to do next, right? But okay, whatever, right? They use the word activate. You can think of it more as this gets the B cell to kind of wake up, right? I like waking up a little bit better, because it doesn't necessarily mean it's going to do something. It just suddenly starts paying attention, right? So this can happen if the B cell is pressed up against bad clown, right? But remember, the nice thing about immunoglobulins is that they don't have to be binding something that's on the surface of another cell. It just has to be something that fits in the Ig arms. So it could be antigen from bad clown, right? And the antigen from bad clown, because it's being shed from bad clown, right? There's a good chances that there are bits and pieces of complement protein stuck to it also. So you can see here, right, the antigen alone carrying a little piece of C3d, right, sticks to the end of the immunoglobulin. But again, the co-receptor can still detect that, right, because it can reach over and bind to the C3d part, and you get that synergistic waking up kind of signal, right? That result in certain genes being turned on in the B cell, and that's the B cell kind of waking up now and paying attention. Hey, what's going on, right? Suddenly it seems like something on my immunoglobulin actually binds. Right? All right, so where is this taking place? This is mainly taking place in lymph nodes. Right, so here we have a zoomed in part of the lymph node. So this would be the part looking at the primary follicle region. Remember, this is where hypermutation happens, but it's also where the B cells that first come into the lymph node are kind of shuttled up there. You know, they're encouraged to go up there because you have these big follicular dendritic cells, the big blue thing in the middle, that's doing what dendritic cells do best, right? They are presenting all kinds of antigen, bits and pieces of things. So you can see here, antigen with little scraps of C3d attached to them are flowing in through the afferent lymphatic vessel into this lymph node. In there, you have other immune cells like macrophages, and macrophages have CR2 on them as well, right? So already macrophages are kind of binding to the C3d, and you have some B cells that came in. Maybe the B cell even came in through the afferent lymphatic vessel because it was in the other lymph node, didn't see anything or whatever, and now it's kind of flowing into this one, right? Or this B cell might have exited the high endothelial venule from the blood and come into the lymph node. But you can see here, it represents, oh, it's immunoglobulins, right, can actually bind to this antigen as well. And the macrophages kind of help the B cells, encourage the B cells to go to the primary follicle region, where the B cells now have a chance to really see a lot of the antigen, because the antigen is being presented in abundance by the big follicular dendritic cell, right? And now these B cells, oh boy, right now they're being told things like, okay, you should go ahead and start multiplying, and hopefully they will then later on get a chance to get instructions from something like a T cell. So let's look at a follicular dendritic cell here. So, these dendritic cells, as I've already described to you, they're pretty remarkable because they have this uncanny ability to really change their shape and morphology depending upon what they are doing, right? The normal everyday dendritic cells that we had been looking at before, as the example of, the little dendritic cells at the site of an infection, like where you stepped on a tack. At first, the dendritic cell will be a certain shape to be able to process proteins, start packing them up. And then the dendritic cell will change its shape to become more of like the delivery truck as it goes through the draining lymph vessel. And then once it gets into a lymph node, right, a typical dendritic cell will just then turn into the salesperson and try to present stuff to things like T cells. Right? Follicular dendritic cells, right, are these much larger dendritic cells that are just kind of hanging out in the primary follicle part of the lymph node to start with, but they also can dramatically change their shape. So here you see a follicular dendritic cell, right? And you're like, wow, this is a messy looking, kind of gross looking cell. Here's the main body of it here. And all this messy stuff is part of the body. These are all protrusions of the cytoplasm, right? These different parts of the cell membrane that are sticking out. And remember, because its job is to present as much antigen and as much different types of antigen as it can, right, it needs to maximize its surface area. So that's why by turning into this big messy thing with lots of almost like tentacles sticking out, that's a lot of surface area on which antigen can be presented. If you zoom in here to this little region here, you're like, what are these lumps, right? That's part of what makes it look kind of gross, right? The lumps. What these lumps are certain regions of the cell membrane of this dendritic cell where you have different proteins that are presenting antigen, like MHC, for example, right? All sort of bound to, maybe larger antigen molecules to present them. And as a result, you have these parts of the cell membrane get kind of pinched together as you have lots of MHC kind of together. And that causes then these funny sort of like little pinched regions of the cell membrane. So don't think it's gross. When you look at it, just think, oh, that's cool, because the reason these parts of the cell membrane are pinched together like this, these are regions where big chunks of antigen or whatever are being presented by many MHC proteins. They're all clustered together on the cell membrane there, and it kind of pulls the cell membrane together there. All right, so let's kind of put this together, right? So that naive B cell that comes in either through the blood or through draining from upstream somewhere, right, inside the lymph node, it has a chance to encounter antigen. And antigen will be presented primarily by follicular dendritic cells up in the primary follicle regions, right? And then those B cells will be kind of like woken up, right? And then they will have a chance to talk to T cells. And those T cells will then give those B cells the proper instructions of what they should do next, right? For example, it'll encourage more of the B cells to replicate, and then it'll encourage some of those B cells to become plasma cells, some of those B cell clones to hang out in the primary follicle and start the formation of germinal centers, right, where the B cells will then undergo hypermutation. So let's talk about this kind of critical B cell T cell pairing up here. One important thing to keep in mind, right, is that T cells, remember, their T cell receptors can only bind small fragments of proteins, peptides, that are presented to them by some other cell. On the other hand, a B cell with its immunoglobulin, it can bind almost any kind of molecule. It doesn't have to be a peptide. That's just like floating by. So in order for a B cell and a T cell to talk to each other, what does that imply? That means the B cell has to be able to present something that the T cell receptor is meant to recognize. So in that case, we are talking about small peptide. Right? And so you say to yourself, well, I don't get it then, because if the immunoglobulin of this B cell is the right shape to bind a carbohydrate or a lipid, right, then how is it that eventually the B cell presents a small peptide? Right? It does so because we tend to oversimplify these cartoons. We always present like antigens as just like a red dot or something like that, right? So we think of it as just one specific type of molecule, when in reality, just like I showed you earlier in the slides today, bits and pieces of antigen that are coming from, let's say, a bacteria, right, that's shedding this stuff, will be made of several different types of molecules all kind of clumped together. Right? So the antigen, right, is actually maybe a collection of some bits of protein, some bits of carbohydrate, maybe some lipid, right? It's just a fragment. So that means a B cell like this one here, its immunoglobulin might recognize the blue part of this dot, right? And let's say the blue part of the dot is, a carbohydrate, right? But part of the antigen has a piece of protein on it also, right? Because after all, this is like a chunk of a bacteria, right? And so that B cell, when it brings in that antigen and processes it, right, the peptide part is what gets loaded onto MHC2, right? Forget about the carbohydrate, the blue part that the immunoglobulin on its surface recognized. After processing this whole antigen, the red dot, which is the peptide part, is what gets loaded up on MHC. And that's how the B cell can interact with a partner T cell. In this case, this is a CD4 helper T cell. And it is, remember, one of 5 different subtypes of CD4 helper T cell. This is the follicular helper subtype. So its job is to hang out in the lymph node and partner up with the B cells to help the B cells know what to do next. Right? And so what happens next is the follicular helper T cell, right? It gets happy because it recognizes the red dot. That's what the T cell receptor recognizes. The CD4 co-receptor confirms that this is MHC2, right? The follicular helper cell, right? It has CD40 ligand on its surface, and the woken up B cell, right, has now started expressing CD40 on its surface so that it can see the CD40 ligand signal. So this B cell knows, right, that this T cell is sending messages that are meant for it. Right? Because remember, that's how this particular T cell is able to essentially say to this particular B cell that it's bound to, hey, these green triangle cytokines that I'm going to send out, these are meant for you and you alone, right? Not another B cell that's in this picture that has a completely different immunoglobulin on its surface and therefore is not a B cell that you would want to activate in this situation. So this is what we call linked recognition between B cells and T cells. And I think it's important to discuss that because otherwise I think it's inevitable that you get confused about how is it that an immunoglobulin on a B cell can bind to a sugar, right? But a T cell can only bind small peptide. So how could it possibly be that these two would actually end up talking to each other? Right? It's because the little sugar that the B cell's immunoglobulin binds to is part of a chunk of something that has things like proteins part of it. Right? So the intersection of B cell and T cell activation is really a super important cornerstone event that is essential for you to have a robust, strong adaptive immune response against something like bad clown. Right? So let's look at this here. naive B cells, naive T cells, they enter this lymph node, right? It could be that this naive B cell and this naive T cell have been floating around together in the blood, and they never even realize that they're sort of like meant for each other, right? Because neither one of them is activated yet. But they come into this lymph node, and in this lymph node, there's evidence of antigen from the site of infection. So the naive B cells, as we said before, they're encouraged to go up to the primary follicle, where they have a chance to really see this antigen being presented by the follicular dendritic cells. Right? Meanwhile, the naive T cells hang out in this light blue area, which we just call the T cell area of the lymph node, where normal, everyday, common dendritic cells have arrived. Right? These are the ones that we'd learned about before that packed up the antigen, trucked it over to the store, and now in the store, this is the salesperson, right, with the B7 badge on it. So once these two cells activate, look what happens, right? These naive B cells, the ones that recognize the red dot, right, their immunoglobulins are happy to pick up the red dot from the follicular dendritic cell. They are now really kind of beginning to activate. Right? They multiply, and they begin to migrate back to the T cell area. Meanwhile, the chosen T cells are busy multiplying, right? And they migrate up to meet the B cells. And this is where they pair up, right? So boy, they didn't know that they were meant for each other, but now, they've paired up, right? Kind of reminds me of like in junior high school, middle school, when you go to dances and it's always so awkward, right? The girls are dancing and the guys were all standing awkwardly on the wall, right? Until you have some of your buddies who start asking the girls to dance, and then you start pairing up, right? So it is like a dance, because what will happen now is a wonderfully kind of choreographed and highly sort of nuanced kind of set of reactions. After these B cells have paired up with a T cell that also recognizes the same antigen, right? Oh, and this is just a reminder again, right? Just like we looked at before with cytotoxic T cells. Remember we said cytotoxic T cells that are out there somewhere binding to cells and killing them. They are able to direct their cytotoxic messages towards the cell they're synapsing with by actually rearranging the internal organelles, like the Golgi complex, the cytoskeleton, etc., to be able to preferentially release cytotoxic cytokines to just kill preferentially the cell that they are bound to. So helper T cells, I mean, look, they're T cells also. They use a similar mechanism, right? So you can see here, this follicular helper T cell that paired up with this B cell over here. This B cell is presenting antigen on its MHC2, and that's what the T cell receptors, TCR, likes. Right? They're held together here by stronger associations, like those molecules like LFA and ICAMs that allow them to really stick together so that these T cell receptors have a chance to really read the MHC. That causes the helper T cell to rearrange its internal protein manufacturing equipment so that preferentially these little green triangles, the cytokines, are sent to the B cell that it's synapsing with, right? So it's not just this big messy cloud of green triangles coming out from this T cell. Why? Because surrounding this pair are lots of other B cells, right? Lots of other B cells that have completely different immunoglobulin, right? That have no business being activated, right? Because that'd be a mess, right? If you activated just any old B cell that happens to be nearby, right? Because then you'd have a whole bunch of totally irrelevant B cells getting activated, right? No. So by doing it this way, this T cell specifically targets that cloud towards this B cell, right? And of course, not shown here, but the other way this T cell is talking to this B cell is through that CD4 T. Oh, no, it is shown. I'm sorry, right over here, CD4 T. So that this B cell knows, hey, these green triangles are meant for you, right? Okay. So when these B cells and T cells pair up in the T cell area, as shown here, some of them begin to migrate toward the medullary region. This is close to the exit of the lymph node, where the helper T cells encourage these B cells to make many, many copies of themselves, right? So they multiply, and they encourage these B cells, hey, become plasma cells, right? Start making your pentameric IgM as antibodies so you can send these out into the blood now, right? So these plasma cells head out into the blood with big clouds of IgM antibody kind of trailing them, right? Being released as your first quick and dirty antibody response against bad clown, right? Because again, up until this moment, you haven't had anything like an antibody that could detect bad clown. Then look, some of these cells then migrate back through the T cell area to the primary follicle, right? So that's why I say it's like a dance, right? These paired cells are going back and forth doing different things. And when they come back here, then the helper T cells encourage those B cells. You guys stick around and start working on making your Ig better, right? So this is where the hypermutation takes place, right? And as we learned last time, as better and better cells get made in here, you're going to get higher and higher quality immunoglobulins, right, through the hypermutation. So then you'd have some pairs of these cells coming back and being told to turn into the next wave of plasma cells that know how to release the improved, more affinity matured versions of the immunoglobulins. Right? So these plasma cells, I've described them as being the sort of endpoint of a B cell's differentiation, right? The B cell, from the day that it was born and it came out of your bone marrow, right? Its dream in life is to eventually be chosen, and to be chosen and turned into a plasma cell so it can release its immunoglobulins and save your life, right? So what do these plasma cells really look like? You know, it's funny when you look at them here, they're drawn as being, oh, they're bigger than a B cell than they started with. And what's all these stripes inside? What's that about, right? Let's take a look. So here is a fully differentiated plasma cell. It is a beast of a cell, right? It is really big. And what are all these little wrinkly things inside? That is all rough endoplasmic reticulum, right? Remember, what's the function of rough ER? Rough ER, is where the ribosomes go to finish making or finish translating proteins, right? So the whole purpose of a plasma cell is just to become a massive antibody factory, right? It's basically, this is it, right? It has no other purpose. So therefore, it almost like rededicates its entire structure to just becoming a massive, massive ball of rough endoplasmic reticulum so that it can pump out, right, hundreds upon hundreds of thousands of individual antibody molecules every minute. Right? And so inside the nucleus, right, the recombined immunoglobulin loci are busy being transcribed, right? The messenger RNA is then being translated by the millions upon millions of ribosomes that are all over this extensive ribosome endoplasmic reticulum network. And even though you can't see it, right, you can imagine this dense cloud of antibody molecules just emanating from this cell, right? So that would be the end of this B cell's life. But it's a happy end for this B cell's life because this particular one, unlike most of its brothers and sisters that came out of the bone marrow, right, they never got this far. This B cell was able to go the distance and eventually become a plasma cell. But at this point, that's it, right? Because the plasma cell at this point cannot turn back into a normal B cell, right? It'll just keep making antibodies until it wears out, and then it's gone. Right? But that's the secret of how your adaptive immune system then releases massive amounts of antibodies in response to infection. All right, let's go ahead and stop.